right, so today we are going to talk about muscle contraction. And in order to do that, we need to talk about how nerves and muscles are related and how they work together to stimulate the muscle cell, how the nerves stimulate the muscle cell and then make it contract. So a skeletal muscle never contracts unless it is actually stimulated by a nerve. If nerve connections are severed or poisoned, a muscle may be paralyzed, meaning no longer will work. Denervation atrophy is the shrinkage of a paralyzed muscle when the connection is not restored. You have somatic motor neurons, and these are nerve cells whose cell bodies are in the brainstem and the spinal cord, and they serve skeletal muscles. You also have somatic motor fibers, and their axons lead to the skeletal muscle. Each nerve fiber branches out to a number of different muscle fibers, and each muscle fiber is supplied by only one motor neuron. So we need to be aware of these definitions. A motor unit is one nerve fiber and all the muscle fibers that are innervated by it. Muscle fibers of one motor unit are dispersed throughout the muscle, all contract in unison, produce a weak contraction over a wide area, provide ability to sustain long-term contraction as motor units take turns contracting, and work effectively to produce a contraction that usually requires the contraction of several motor units at once. In an average motor unit, there are 200 muscle fibers for each motor unit. Small motor units have a fine degree of control, and there are 3 to 6 muscle fibers per neuron. We find these in the eyes and the hands. There are also large motor units, and these have more strength than control. They are powerful contractions supplied by large motor units, such as the gastrocnemius, which is in your calf. And here we find about 1,000 motor fibers per neuron. So there are many muscle fibers per motor unit. There is something called the neuromuscular junction, and we're going to talk about this extensively today. A synapse is the point where a nerve fiber meets its target cell. And this NMJ, or what is called a neuromuscular junction, is when the target cell is a muscle fiber. Each terminal branch of the nerve fiber within the neuromuscular junction forms separate synapses with the muscle fiber. One nerve fiber stimulates the muscle fiber at several points within that neuromuscular junction. So, there are several different components that make up this neuromuscular junction. There is a synaptic knob. This is the swollen end of the nerve fiber, and it contains synaptic vesicles that are filled with acetylcholine, abbreviated as A, CH. There is what's called a synaptic cleft, and this is a tiny gap between the synaptic bulb and the muscle sarcolemma, which remember is the outside of the muscle cell. Then you have a Schwann cell. This envelopes and isolates all of the neuromuscular junctions from surrounding tissue fluid. And we see exocytosis happening here because the synaptic vesicles are releasing this acetylcholine into the synaptic cleft using exocytosis. There can be 50 million acetylcholine receptors. These are proteins incorporated into muscle cells, the muscle cell of the plasma membrane, that sarcolemma. So there can be 50 million. And then there are junctional folds of that sarcolemma beneath the synaptic bulb, and this increases the surface area and can then hold more ACH receptors. If you are lacking receptors, you could have paralysis. And this is what we see in a specific disease called myasthenia gravis. And this is a type of paralysis of the muscles. There is also a basal lamina, which is a thin layer of collagen and glycoproteins that separates the Schwann cell and the entire muscle from the surrounding tissues. These contain acetylcholinesterase. ASE on the end of that means it is an enzyme, and this actually breaks down the acetylcholine after contraction, which then causes relaxation. So once the contraction is done, you have to get rid of the acetylcholine, and you do that by using this enzyme. It comes in and breaks it down so that the contraction is no longer taking place, and therefore the muscle would relax. Okay, there are certain neuromuscular toxins. There are things that can interfere with your muscles. And this is because they interfere with the synaptic function, and therefore they can cause paralysis of the muscles. There are things like pesticides that can do this. And depending on the types of things they may include, they can inhibit or block your receptors. So here's one that can actually bond, and sometimes in pesticides we see a chemical that can actually bind to that enzyme that's supposed to break down the acetylcholine. If that enzyme can't get loose or can't break down the acetylcholine, basically your muscle would never stop contracting, right? Because that enzyme needs to break it down so that relaxation can occur. But if it can't degrade that acetylcholine, you can have what it's saying here, a spastic paralysis, a state of continual contraction. This could cause suffocation, depending on which muscles are continuously contracting. You may have heard of something called tetanus or lockjaw, and that's kind of the same thing here as what we're talking about. When these different toxins can get in and block these receptors, it can cause our muscles to not work properly. So glycine in the spinal cord normally stops motor neurons from producing unwanted muscle contractions, but a tetanus toxin blocks those glycines that are released in the spinal cord and causes then an overstimulation and spastic paralysis of the muscles. You can also have a flaccid paralysis, and this is a state in which the muscles are limp and cannot contract. Curare competes with acetylcholine for receptor sites, but it does not stimulate the muscles. This is a plant poison used in South America in blowgun darts. I believe it also is in some snake venoms. And then you have botulism. This is a type of food poisoning caused by a neuromuscular toxin secreted by a special bacterium, Clostridium botulinum. And this is what we see in Botox, which basically blocks the release of ACH and causes flaccid paralysis, which is why Botox will stop your wrinkles, because it's literally deadening the nerve that is causing the wrinkle to appear. Okay, muscle fibers and neurons are electrically excitable cells. Their plasma membrane exhibits voltage changes in response to stimulation. Electrophysiology is the study of the electrical activity of cells. So, couple different things happening. In an unstimulated or resting cell, there are more anions, meaning negative ions, on the inside of the plasma membrane than there are on the outside. This means that the plasma membrane is electrically polarized or charged. There are excess sodium ions in the extracellular fluid, while there are an excess of potassium ions in the intracellular fluid. Also, in the intracellular fluid, there are anions such as proteins, nucleic acids, and phosphates that cannot penetrate the plasma membrane. These anions make the inside of the plasma membrane negatively charged by comparison to the outer surface. We have what is called a voltage or an electrical potential, and this is a difference in electrical charge from one point to another. Your resting membrane potential is about negative 90 millivolts. This is in a resting cell, and this value, this voltage, is maintained by what we call the sodium-potassium pump. Now, once we have a stimulated or active muscle fiber or nerve cell, things change. The ion gates open in the plasma membrane. The sodium instantly diffuses down its concentration gradient into the cell. These cations override the negative charges in the ICF, and something called depolarization happens. And this means that the inside of the plasma membrane becomes briefly positive. This causes an immediate reaction. Sodium gates close, potassium gates open, and potassium rushes out of the cell. It is repelled by the positive sodium charge, and partly because of its concentration gradient. The loss of these positive potassium ions turns the membrane negative again, which is called repolarization. An action potential is a quick up and down voltage shift from the negative resting membrane potential to a positive value and back to that negative value again. And within that short time, all these things I just mentioned happens. A resting membrane potential is a stable voltage seen in a waiting muscle or nervous cell. It has not been stimulated yet. Whereas the action potential is a quickly fluctuating voltage seen in an active stimulated cell. An action potential at one point on a plasma membrane causes another one to happen immediately in front of it, which triggers another one a little further along, and so on and so on and so on. It's a chain reaction that happens. There are 4 major phases of contraction and relaxation. The first phase is excitation. This is the process in which nerve action potentials lead to muscle action potentials. We then have what's called an excitation-contraction coupling, and these are the events that link the action potentials on the sarcolemma to activation of the myofilaments, thereby preparing them to contract. We then have the active contraction, which is a step in which the muscle fiber develops tension and may shorten. And then we have relaxation. When the work is done, the muscle fiber relaxes and returns to its original resting length. So now we're going to look at those steps in depth. Here you have a picture. You have lots going on. You see your synaptic knob here. You can see the motor nerve fiber coming in. The nerve signal is designated by the red arrow. You see the sarcolemma. You see the infolds. You see all the things we just discussed. So the first step in excitation is that a nerve signal is arriving and basically saying, hey, we're ready to stimulate. So the nerve signal opens the voltage-gated calcium channels in the synaptic knob. Calcium stimulates excitosis of acetylcholine from the synaptic vesicles. So the calcium is released. The calcium is causing the acetylcholine to be released into the synaptic cleft. This is steps 1 and 2 of excitation. The signal arrives. This opens up the voltage-gated channels. Calcium is able to be released and stimulate the acetylcholine to be released into the synaptic. It's released in synaptic vesicles into the cleft. Why? Because we need that acetylcholine to bind to the receptor. The binding of this will cause something else to happen. So when we have the acetylcholine molecules binding to each receptor, we have an opening of the sodium and the potassium channels, which is what you're seeing in this number 4 picture. You can see how the Na is moving in one direction and the potassium is moving in the other. So the Na, the sodium, enters, shifting that resting membrane potential from a -90 that we spoke about to a positive 75. This then causes the potassium to exit, and this then reverses that resting membrane potential back to its -90. This quick voltage shift is called an end plate potential, an EPP. Step 5. We now have the opening of the voltage-gated regulated ion gates. And this causes what's called a creation of these action potentials. So now we have this voltage change in the end plate region, which opens up these voltage-gated channels and produces what's called an action potential. And this action potential is going to spread over the entire surface of the muscle. So, once we have that action potential, we have what's called an excitation-contraction coupling. We need that action potential to stimulate the contraction. So in step 6, we have this action potential being propagated down these T tubules. The T tubules are just these infolds in the sarcolemma that allow for penetration into the muscle. So this causes an opening of the voltage-gated ion channels in the T tubules and the calcium channels in the SR, the sarcoplasmic reticulum. We now have calcium being able to enter the cytosol, which is number 7. Calcium is released from the terminal cisternae into the cytosol. Once calcium is free, it is able to bind to troponin on the actin or the thin filaments. And once the troponin is able to bind, it causes what's called a troponin-tropomyosin complex, which then changes the shape and exposes the active sites on the actin. So it's literally pulling off those tropomyosin cords, the green if you see there, and exposing the active sites on the actin, which are the red little beads here. And in picture 9, you can now see that the active sites are open. We need those open because the myosin head is going to fit into those. Contraction. Now we have the myosin ATPase, which is an enzyme. Remember, ASE again, always an enzyme. In the myosin head is going to hydrolyze an ATP molecule, which is what's happening here in number 10. So we are breaking off one of those P's, right? We have ATP triphosphate. When one is broken off, we have ADP diphosphate. And that extra energy allows for the activation of the head, or what we call like a cocking, which means it's being clicked back so that it is now able to connect. In number 11 here, you see basically what we call the myosin-actin cross bridge. And this happens because the head of the myosin is now binding to the actin, those active sites, and it's forming this cross bridge. It's forming a connection. Then we see the myosin head releasing the ADP and the P. It flexes, pulling the thin filament past the thick filament, which is what we call a power stroke. Upon binding more ATP, myosin releases the actin and the process is repeated. Each head can perform 5 power strokes per second. Each stroke utilizes 1 molecule of ATP. This is muscle contraction at a molecular level. Then we need to relax, right? After the contraction is performed, we need our muscles to relax. So the nerve stimulation and the acetylcholine release needs to stop. We are no longer being told that we need that muscle to contract. We now have the acetylcholine esterase coming in, breaking down the excess acetylcholine, and the fragments are reabsorbed into the synaptic cleft, right? We have to get rid of the extra ACH because we no longer need it, because we are no longer contracting. We are now relaxing. Once that happens, The extra calcium is pumped back into the SR by active transport, and the calcium binds to the calquestrian while in storage in the SR. ATP is needed for muscle relaxation as well as it is needed for contraction, because there's lots of different things going on. So the calcium is removed from the troponin. It is pumped back to the SR. The tropomyosin then blocks the active sites. It moves back over to block those active sites. Therefore, the muscle fiber ceases to produce or maintain any tension. The muscle fiber now returns to its resting length because it is relaxed. So it's just the opposite of what we saw going into the contraction. Everything is just reversed, and now the muscle can relax. Have you ever heard of rigor mortis? Well, that is a hardening of muscles and stiffening of your body beginning 3 to 4 hours after someone dies. And this is due to a deteriorating SR and the release of calcium. So once you have a deteriorating sarcolemma, this allows the calcium to enter the cytosol, meaning that calcium can activate the myosin-actin cross-bridging, and it can allow these muscles to contract but never to relax. Because as we said, muscle relaxation requires ATP just like the contraction does. But there is no longer any ATP production in someone that is no longer living. So the muscles have the ability to contract, but not to relax. So rigor mortis will peak about 12 hours after death, and then it will diminish over the next 48 to 60 hours because then the body just starts to break down. So, I know that was a lot of information, but please listen again if you have to. Read over your text, look at the notes. We do need to be aware of the steps that we just went over. From the very beginning of when the cell is excited, to the contraction, to the relaxing. Please let me know if you have any questions.